Up until this point in the semester, we've been focusing on the things that happen inside of cells, but we haven't really spoken about what drives those processes or what causes those processes to initiate. We're going to diverge a little bit here over the next couple of class periods to talk about the induction of those intracellular processes and specifically what induces those processes in a multicellular organism like ourselves. In order for a multicellular organism to function properly, all of the things that happen inside an individual cell must be coordinated with the things that happen in other cells. There are things that have to occur in order for those processes to be coordinated, and one of those is intercellular communication. In order for a cell to simply survive, it must receive signals from the extracellular environment. Just like ourselves, in the absence of any communication, a cell will die. There are multiple signals that can be received in order to induce a cell to survive. But if a cell receives additional signals, it can do other things. For example, in this image, the cell is receiving survival information as well as additional signals that induce the cell to grow, copy its DNA, and ultimately to divide. Alternatively, other signals can be received so that in addition to the survival signals, we have in this image other inputs that cause a cell not to grow and divide, but instead to differentiate into some other terminal cell type. In order for a cell to communicate, there are two parts of that communication. One of those is the incoming signal, and the other is the way that signal is received or heard. I want to speak about each of these two components of communication, the signal and the receptor, independently. First, focusing upon the signal. The path that a signal takes dictates the type of communication that occurs. Endocrine communication depends upon the signal traveling through the bloodstream. Now, the distance that that signal travels will certainly vary. There are examples where traveling through the bloodstream is a very short distance, for example, from the hypothalamus to the pituitary gland. But it is still endocrine signaling because the signaling molecule, in this case, the endocrine hormone, is released into the bloodstream and carried to a target cell through that bloodstream. If the signal is released into the extracellular environment, this type of communication is paracrine communication. In paracrine communication, again, the signal is released into the extracellular space, diffuses through that extracellular space, and then when it finds a cell that is able to receive the signal, it induces a response. You'll notice in paracrine communication, the cells that are communicating are of different types. And we note that because the signaling cell has a brown nucleus and the receiving cell has a blue nucleus. It is possible for a cell to communicate with another cell of its same type. In this case, we call the communication autocrine communication. the signaling molecule is still released into the local environment and diffuses as far as it can within that local environment to communicate with a nearby or neighboring cell. Note that in autocrine communication, since all of these cells are of the same type as indicated by their blue nuclei, it is possible for a cell to talk to itself. In other words, the signaling molecule that is released into the local environment can bind to a receptor on a cell that is of the same type in the local vicinity, or it could bind to a receptor on the cell that releases the signal. The point here is that the signaling molecule is released into the extracellular space and is able to affect nearby cells within diffusion distance. Another type of communication is synaptic communication, and that is where a signaling molecule is released into a synapse so that it can affect a very nearby neighboring cell. We won't spend much time talking about synaptic communication because we've already done that in chapter 11. Finally, a fourth type of communication is called contact communication or contact-dependent communication. And another name to describe this, or another word to describe this, is juxtacrine communication. In juxtacrine communication, the signaling molecule is not released. Rather, it is membrane bound, and a cell that is next door or adjacent to the communicating cell is the only cell that might possibly receive the signal. It must have a receptor for that membrane-bound signaling molecule. Here are some examples of signaling molecules. Those shown in this table are endocrine signaling molecules. You'll note that some of these endocrine signaling molecules are steroid hormones like cortisol and estradiol. Others are proteins like insulin, and others are amino acid derivatives like epinephrine and thyroid hormone. So the type or the chemical characteristic of the signaling molecule is one parameter that we might consider. And again, in the previous slide, another parameter of signaling molecules is the path that they take. Here we have a table showing you some examples of other signaling molecules. Paracrine or autocrine signaling molecules are those that are released into the extracellular environment. Shown here are some examples of protein signaling molecules, various growth factors, for example, of amino acid derivatives, histamine, for example, or even of gases, nitric oxide, that is released into the extracellular space and diffuses to adjacent cells. Examples of synaptic signaling molecules are acetylcholine or GABA. And one example of a juxtacrine signaling molecule is delta, the transmembrane protein that interacts with the Notch receptor on adjacent cells. Before talking about the receptors, I want to briefly talk about the response that a cell might have when a signaling molecule is bound to a receptor. Let's talk about a single signaling molecule, acetylcholine. Acetylcholine can induce a number of different cellular responses dependent upon the cell type. Here we have 3 different cell types shown. A cardiac pacemaker cell that will depolarize at a decreased rate in response to acetylcholine. A salivary gland cell that will release vesicular contents upon acetylcholine signaling, or a skeletal muscle cell that will contract in response to acetylcholine. How is it possible to get each of these different cellular responses to the same type of signaling molecule? One way that can occur is if we look at the salivary gland cell and the skeletal muscle cell carefully, we see that these two cell types have different acetylcholine receptors. In the skeletal muscle cell, we have an ion channel, and in the salivary gland cell, we have a multi-pass transmembrane protein. Each of these is an acetylcholine receptor, but it's a different type of acetylcholine receptor. In response to acetylcholine binding, then, these two different cell types are going to have different responses. Comparing the cardiac pacemaker cell and the salivary gland cell, we see that each of these cell types has the same type of acetylcholine receptor, that multi-pass transmembrane protein. However, different intracellular events can induce the different responses. We'll spend a lot of time talking about these different intracellular events next time. Regardless of the intracellular events that occur, sometimes the response is very fast and sometimes the response is slow. When a cell has a fast response, it's typically a very short-term response, meaning that the effects are short-term. And those short-term fast responses are due to altered protein activity inside of the cell. Alternatively, when a cell responds slowly to some type of communication signal, that slow response is because different proteins have to be made, and that altered protein synthesis is the reason the response is slow. However, because different proteins are made, the response lasts much longer, so that a slow response is typically associated with longer-term effects. Now let's talk about the receptor. The previous images have shown you receptors that are on the extracellular face of the cell. These are cell surface receptors, and cell surface receptors are required when hydrophilic signaling molecules are used. Hydrophilic signaling molecules are unable to pass through the membrane, so they must bind to an extracellular portion of a transmembrane receptor in order to induce some sort of response inside of the cell. Alternatively, we know that some signaling molecules are hydrophobic. Examples might be testosterone or estradiol. These signaling molecules are hydrophobic, so they are able to freely pass through the membrane. In this case, the receptor for these hydrophobic signaling molecules is intracellular. When the hydrophobic signaling molecule binds to an intracellular receptor, it will induce changes in cellular behavior. Let's look at the intracellular events that occur when a hydrophobic signaling molecule binds to an intracellular receptor. Again, here are some examples of hydrophobic signaling molecules: cortisol, testosterone, thyroid hormone, and even the gas nitric oxide. These hydrophobic signaling molecules diffuse through the membrane and bind to an intracellular receptor to elicit a cellular response. What I'm saying is that these signaling molecules can diffuse into any cell. However, it's only those cells that have the receptor specific to that signaling molecule that are going to be able to respond. The type of response that will occur upon intracellular receptor binding is going to depend upon the type of intracellular receptor. We'll talk about two of those. The first type of intracellular receptor is a receptor transcription factor. One example of this is steroid hormone receptors. Here, cortisol is able to freely diffuse through the cell membrane and binds to the receptor transcription factor in the cytoplasm. Cortisol binding to the receptor in the cytoplasm induces a conformational change in that receptor such that a nuclear localization signal is exposed. That nuclear localization signal is used to transport the receptor hormone complex to the nucleus, where that complex binds to a regulatory region on DNA, specifically an activator site that induces transcription of target genes, cortisol responsive genes in this case. We're talking about altering gene expression in response to steroid hormone binding. And that response is a slow response because it takes a while for genes to be transcribed, proteins to be translated, modified, and induce their responses. However, these are going to induce long-term effects, again, in response to a steroid hormone binding to a receptor transcription factor in the cytoplasm. Faster responses can occur, for example, when the gas nitric oxide is released into the extracellular space. This occurs in a blood vessel, and the blood vessel is shown at the top left. There are two cell types that we'll concern ourselves with. One is the endothelial cell, which is a specialized epithelial cell that lines the blood vessel. And the other is the smooth muscle cell that lies adjacent to it in sort of the middle of the blood vessel. In response to acetylcholine, the endothelial cell will activate an enzyme called nitric oxide synthase. Nitric oxide synthase induces the production of nitric oxide that is able to freely diffuse through the endothelial cell membrane into the extracellular space. It is able to diffuse then into the smooth muscle cell, and it is the smooth muscle cell that is going to respond to nitric oxide bound to a receptor enzyme in the cytoplasm. The enzyme specifically to which nitric oxide binds is called guanylyl cyclase. Guanylyl cyclase converts GTP to something called cyclic GMP, guanosine monophosphate, and the phosphate sort of circles around such that it links the oxygen that's associated with the number 3 carbon to the oxygen that's associated to the number 5 carbon. Cyclic GMP production in the cytoplasm of the smooth muscle cell induces relaxation, and relaxation causes vasodilation. In other words, the blood vessel relaxes and gets larger in diameter. An enzyme known as phosphodiesterase is able to cleave cyclic GMP such that it is no longer cyclic, creating GMP. GMP has no intracellular response, and therefore a smooth muscle cell would contract when phosphodiesterase is active. Viagra is a pharmaceutical drug that inhibits phosphodiesterase, thereby extending the lifespan of cyclic GMP in the smooth muscle cell and extending the duration that the blood vessel is dilated. I'll let you hypothesize as to which blood vessels specifically this might be targeted. This response, again, is fast, and because it is fast, it is typically short-term in duration, unless, of course, we take Viagra. Signaling through cell surface receptors induces a cascade of effects inside of the cell. Let's look at those. Here are some examples of the hydrophilic signaling molecules that will bind to cell surface receptors. They can be small polar molecules like histamine, an example of peach lactone, the smell that peaches have. We also have proteins like epidermal growth factor and insulin, and signaling molecules like acetylcholine. These bind to cell surface receptors because they are unable to pass through the plasma membrane. The cell surface receptor, in response to signal molecule binding, will change shape, and that shape change induces a number of effects inside the cytoplasm that ultimately will induce a change in cellular behavior. This depends upon the activation or inactivation of intracellular signaling molecules that we'll term relay molecules, or more commonly, second messengers. Here we see an example of a typical intracellular cascade that results when a hydrophilic signaling molecule binds to a cell surface receptor and induces a shape change such that intracellular signaling molecules that we term second messengers become activated. Each of these second messengers can induce the activity of effector proteins that will induce target cell responses. Looking at a slightly less simplistic image of a signaling cascade, we see that there are a number of steps in the cascade, and we might wonder why the complexity. Why can't an extracellular signal binding to a transmembrane protein allow the transmembrane protein's shape change to directly induce the activity of one of these effector proteins such that cellular behavior would be altered? There are a couple of answers to this question. The first is that with these multiple steps, we have the opportunity to amplify the signal. That is because a single extracellular signaling molecule binding to a receptor will ultimately activate a number of second messenger molecules, and each of these second messenger molecules can subsequently activate downstream effector proteins such that the cellular response is amplified, meaning that a single signaling molecule can induce a pretty large response in a target cell. Having multiple steps along the cascade pathway also allows us a number of opportunities to modulate the effect. And by modulate, I mean either amplify or inhibit. Having each step with the ability to modulate the effect allows us to fine-tune cellular responses. For example, if in response to this extracellular signaling molecule binding, a cell received other signals such that altered metabolism wasn't desired, we could interfere with some step that is responsible for altering metabolism and interfere with altered metabolism, but not interfere with altering the cell shape or altering gene expression. Again, we use this signaling cascade mechanism upon binding to a cell surface receptor because it affords us the opportunity to amplify the signal and or modulate the signal at multiple steps so that we can get the desired cellular response. This whole process of using a cascade is referred to as signal transduction. Signal transduction is what occurs when a signaling molecule binds to a cell surface receptor and induces an intracellular cascade that ultimately alters cellular behavior. Because these signal cascades depend upon molecular binding to activate second messengers, I want to spend just a little bit of time calling attention to some common protein domains that are involved in molecular binding in these signaling cascades. One of those is an SH2 domain. An SH2 domain is a protein domain that is responsible for binding to a phosphorylated tyrosine on some other protein. The other starred protein domain at the bottom of this table is a phosphotyrosine binding domain, or a PTB domain. This also binds to phosphorylated tyrosines. This should signal to you that common to many of these cascades is going to be phosphorylation of tyrosine amino acids. In addition to SH2 and PTB domains, a couple of other domains that we'll see. One is an SH3 domain. SH3 domains are responsible for binding to polyproline sequences on proteins. These polyproline sequences form a three-dimensional shape that is recognized by an SH3 domain in a binding partner. And finally, a plextrin homology domain is responsible for binding to a phosphorylated inositol head on a phosphatidylinositol lipid in the membrane. Let's look at how each of these is going to work. Here we have a transmembrane protein that has become activated in response to extracellular signal binding. That activation results from phosphorylation on a tyrosine amino acid, and that phosphorylated tyrosine is recognized by a phosphotyrosine binding, or PTB domain, on an adapter protein. That adapter protein also happens to have a couple of other components. One of those is a plextrin homology domain that can bind to a phosphorylated inositol protein. The other are multiple sites that can be phosphorylated by the activated receptor, and those phosphorylated tyrosines on the adapter protein can be recognized by the SH2 domains of a second adapter protein. This second adapter protein also happens to have a couple of SH3 domains that will recognize polyproline sequences in a couple of other proteins. One, an effector enzyme, and two, a scaffold protein. In this way, the cascade can occur in a stepwise fashion: activation of the receptor, activation of adapter protein 1 upon binding, activation of adapter protein 2 upon binding to adapter protein 1, and then activation of enzyme and activation of scaffold protein by molecular binding to adapter protein 2. Hopefully you see how this cascade might work. Relay proteins aren't only activated by molecular binding. I just want to remind you that there are a couple of other ways that we can activate proteins. One of those is by phosphorylation, and we'll see phosphorylation a lot in these signaling cascades. Specifically, phosphorylation of tyrosines, as we've already noted, by the SH2 and PTB domain being very common. And the other is signaling by GTP binding proteins, or G proteins, that are able to not only bind GTP and become activated by GTP binding, but are also able to hydrolyze GTP to GDP and thereby turn themselves off. Finally, these cell surface receptors about which we've been speaking come in 3 flavors. First, our ion channel receptors. We've referred to these as ligand-gated channels in previous lectures. And because we've spoken about these previously, we won't spend a lot of time talking about them here. We will, however, spend a fair bit of time talking about both G protein coupled receptors and enzyme coupled receptors, starting with the latter. These enzyme coupled receptors that we're going to focus on anyway are referred to as receptor tyrosine kinases, and these are very common cell surface receptors that induce signaling cascades. We'll learn about these in subsequent videos.